Thank you very much, Steve and Jeanette, for inviting me to talk here in this workshop. So I'm at the University of Toronto in the Donnelly Center building, which is a newer interdisciplinary research building on their university campus. And I'm going to talk about network visualization and analysis today. And then tomorrow I'll talk more about some specific analyses that we've done with network tools and add-ons that we've additional features of Cytoscape. So today's more basic and maybe a little bit more general than what's been going on today, but I think it's definitely in the same line of thinking. And it's sort of, I think I heard this from Steve earlier, when you're doing a network analysis, you want to think about the paper, and in the Cytoscape way of thinking about it, Cytoscape is a tool for loading network data and visualizing it, and you can load in lots of additional types of information. And then there's a lot of different tools for analysis and visualization. And then one of the things that people like to do with Cytoscape is prepare a visualization for a figure in a paper where you want to visualize a network. And there's Nature Protocols that we wrote quite a few years ago now already that goes through one specific example relating to gene expression data analysis. So this lecture, I wanted to just give some general, present some general ideas about network visualization, mostly. And then I'll talk about network analysis, but I'll just go through some examples of different types of network analyses that will that you can do with Cytoscape and other tools. So I think we don't some of this may be a little bit repetitive from the previous lectures today, but networks generally represent, I'll just talk about it from my viewpoint. Networks in biology represent networks represent relationships, and in biology, these relationships are typically physical relationships like protein interaction, regulatory, transcription factor regulates these genes, genetic interactions, which are more gene-gene interactions, and functional interactions, which I think the correlations between genes from gene expression profiles that we talk about mostly in this course are a type of, and people use that to predict gene function, for instance. So, and in general, one of the most useful things about networks is that they're very useful for discovering relationships in large data sets. So they're much better than tables in Excel. If you just had a bunch of relationships between genes or samples or anything, if you plotted it in table format, it wouldn't be very useful because it'd be difficult to kind of see how global relationships work. But they're also extremely useful to visualize lots of different types of data together and see interesting patterns, and also, of course, for network analysis. So here's an example of a network that was visualized with Cytoscape with a lot of different types of information overlaid. The circles here, the nodes represent genes, happen to be from yeast, and the edges, the links or interactions happen to be protein-protein interactions. And then the thickness of the edges are proportional to the correlation of gene expression profiles in an experiment that was profiling cell cycles stage in yeast. And then the size of the circles, the nodes, are proportional to the amplitude of the transcription across the cell cycle. So the big circles mean that the genes were expressed at a high level at some point in the cell cycle. And then the colors of the nodes represent functions, or in this case, locations in important complexes in a set of genes. The kinetochore is blue, the nucleosome is green, the replication fork is red, and yellow is other functions that aren't specified. So you can see that there's quite a lot of information on this network, and I'll go into a little bit more detail about what you kind of want to extract from a network like this in later slides. We also in a lot of times when people are thinking about networks, a lot of what was presented today is modules of networks, but there was also some mention about pathways, KEGG pathways, and there's some relationship between biological pathways and networks. So networks are kind of a more general type of idea, and pathways are more specific type of relationship between proteins, for instance, that involve protein interactions, or regulatory pathways that involve transcription factors regulating other genes. And so a lot of these types of pathways are networks, but they have a lot richer type of information, and they also have some information about often causality in the system, whereas networks usually say that just two things are related. You don't always know a direction on the arrow on the edge. Okay, so why are networks useful? I think we've already established this in the course today, but I just want to give one more example. Did you talk about the 6 degrees of separation earlier? So probably most people in the room have heard of this idea of 6 degrees of separation. How many people have heard of that? Okay, so many people. So this is the idea that everyone in the world is connected to everyone else with at most 6 hops through a friendship network or an acquaintance network. And this was pioneered, this idea was sort of described originally by Stanley Milgram, who did some experiments in the 60s where he took some people in the middle of the country like Nevada or Wyoming or something and asked them to figure out, to send postcards to someone in Boston. They only knew the person's name and some basic information about that person, and they didn't know the person's address, and they had to send the message via their friends. So they had to send it to someone who they knew on a first name basis. So people sent postcards to their friends that they thought were closer to somebody in Boston. And at the same time, they would send a postcard back to the researchers who could then track where the postcards were going. And through this, it turned out that on average, it took 6 hops to reach somebody in this random person in Boston. So that's kind of an interesting idea, but the real question here was, they're mapping out a network of friendship relationships, and the real question was, which path should you take through this network? And these days we've mapped out our social networks in huge amount of detail on Facebook and LinkedIn, and you might want to, and we also have a lot of networks in biology. You might want to know how 2 nodes are connected in these networks. And this, in the biological situation, do you have, if you're looking at a big protein-protein interaction network, are 2 nodes connected? And if so, how? It turns out that in computer science, people have developed a lot of, many of you might know this from the field of graph theory. People have developed a lot of algorithms that work with networks, and in computer science they call them graphs. And one of the basic algorithms is answers this question. If 2 nodes are connected, it will tell you that they're connected. If they're not connected, it will tell you they're not connected. And if they're connected, it will find the shortest path between them. And that algorithm is called shortest path by breadth first search. And the interesting thing about it is that computer scientists a long time ago proved that this would answer the question optimally and perfectly all the time, and you'll always get the correct answer. So it's nice to know that you can take these algorithms from computer science that have been rigorously proven mathematically and just apply them to answer questions. And that's some of the one of the main, I think, an important reason why network analysis has become popular, because there's a lot of interesting algorithms that you can just apply to questions in biology. So yes, it will find if 2 proteins are connected, and if so, how. One question about this is if is this the shortest path between 2 proteins in a protein interaction network biologically relevant? It may be, but it may also, you may not have all the information about the context, whether the proteins are expressed at the same time and the same place and in the same cells, and so you might need additional information to get a better understanding, but it's still useful. Okay, so just a few examples that come from the world of Cytoscape. Each one of these examples has a feature in Cytoscape that allows you to do this type of analysis. People have used networks in biology to predict gene function. If you don't know the function of a gene, you can find out what its neighbors are, and that might tell you something about the function. Maybe that gene is connected to only genes in the cell cycle, and so maybe it's also involved in the cell cycle. People have found modules in networks, as we've talked about earlier. people and depending on the network, there might be different types of those modules might mean different things. So in a protein interaction network, those modules often indicate protein complexes, whereas in a correlation network, they're more the modules that were discussed earlier today. People have studied network evolution, people have predicted new interactions and functional associations. When I say functional associations, it's sort of a relationship between genes or proteins that allows you to make that basically says that those genes are functionally related, whether they're sequence similar or they interact physically or they're correlated in many gene expression experiments. Those are all give you some clue about the function of a gene. There's also quite a few interesting applications of network analysis in disease. Identification of disease subnetworks, whatever those networks might be. Often, from my perspective, people have been looking at protein interaction networks and looking to see, to find modules in these protein interaction networks that are correlated with subtypes of disease. And you might be able to use this for diagnosis as well. And also genome-wide association studies using networks. Okay, so it's important to understand, at least from the network view, when you're viewing a network, what's missing. So typically, networks that we see are often static. You might see networks at different time points, which is useful, but it's difficult to represent real dynamics that we know are happening in the system, in the cell or the physiology. There are there's a large community of researchers who work with data at that level, using more detailed mathematical representations to model dynamic processes. And typically in network analysis, we don't think about things in that way, but one of the reasons that we don't is we don't usually have, often when we're working with networks, we're working with large networks and we want to say something about all the genes in the genome. We're looking at genomic information, we're interpreting genomic information, and when we do that, we don't have, when we're interested in that level of breadth, we don't have kinetic constants for every protein, and that will help allow us to do this kind of modeling. There's also detail missing, so we often represent genes as nodes, but we know that genes have a lot of attributes. They have structure, they have exons, introns, they have domains on the protein level, and a lot of that is not represented. And then there's context, what cell type and developmental stage the gene is expressed in might not also be represented. So just important to realize that. Okay, so just to quickly summarize the first very basic part, that networks are primarily useful, I think, for seeing as a visualization tool for seeing relationships in large data sets. But it's very important to understand what the nodes and edges mean. So when you see a network, the nodes could mean different things. For instance, Steve mentioned earlier, you could have nodes representing samples, patients, or genes. So if someone gives you a network, you can't just assume that it's a gene correlation network. You have to ask what the nodes and edges mean. And there's also a bewildering amount of network analysis techniques that are out there. And so it's important to kind of define the biological, to know exactly what you want to do with your large network or gene list or gene expression data. Do you want to do diagnosis? Do you want to find new modules? Do you want to find new pathways? And there may be some methods out there of network analysis methods that help you do what you want to answer that question. So sort of two ways of doing that. One is sort of you have the question and you ask, you find out if there's a solution. You can ask an expert or a colleague, or at a course like this, put up your hand and ask. Or the alternative is get to know many methods and see how they can be applied on your data. So that's more of the expert view. Okay, so I'm next going to talk about network visualization and just talk about some basics of network visualization. So there are three major types of ways of representing networks. The simplest way is just a list of relationships, This is not very useful from a network visualization point of view. We've seen these other two in the course today. Sort of standard way of representing networks is nodes and edges. So circles are nodes and lines are edges. Lines can have edges can have weight. Nodes can have other properties like the number of connections is called the degree, and the amount of connections in a little area or in the neighborhood of a node divided by the number of possible connections it could have is called the clustering coefficient, basically, and sort of represents the density of the network around that node. And you can have direction as well on these edges. The equivalent representation is this adjacency table or adjacency matrix view, often visualized as a heat map, where you show a color based on the weight of these edges. So stronger weights here are colored, stronger color here. In these different representations, there's a couple of example edges that are shown as they're colored that are equivalent throughout. So A3 to A5 connection, the weight of 1 is shown here, and it's shown as a box here and here. So in this, I think we covered this before, but in this representation, you have all the nodes on one side and all the nodes, the same nodes on the other side, and you have a number or a color in the where if there's an edge between nodes on this side and nodes on this side. And if you have undirected edges, you will get a symmetric. If you have an undirected network, you'll get a symmetric matrix. If you have direction, then you'll only have a color or a weight on one side of this triangle here, on the lower triangle or the upper triangle. So, so the reason why people would visualize using one of these two methods is they sort of have different strengths and weaknesses. Does anyone know why you'd want to visualize a particular data set as a network or a heat map? Any guesses? So anyone? No? Even the experts? So basically, a network is more useful when the data is sparser. So when there are fewer connections between genes. If you had all the genes connected to all the other genes, the network would just be a big connection. Everything's connected to everything else. It would be difficult to see any structure in the network. In that case, this heat map is much more useful. But in this case, this network is fairly sparse, and this heat map uses up a lot of space just to show no connections. And so it really depends on your data set. If you have a fully connected network, then probably best to show it as a heat map. Okay, so the next thing I want to talk about, the next basic idea, is automatic network layout. So this is a requirement of network visualization. If you didn't have automatic network layout, your networks would look like this, which are affectionately called hairballs by most biologists when they see this. Although somebody told me it looked like the Death Star or something like that. But so if you were just drew your nodes and drew all your connections, you get something like this. So fortunately, there are many different algorithms for laying out networks automatically, and the one thing that all these algorithms have in common is they try to reduce overlap between of nodes and edges. So if you can, because people recognize that if you have nodes that are on top of each other, you can't see the nodes, right? And if you have edges that are crossing, it creates a lot of complexity. It's hard to trace where one edge goes from one place to the other. So just those two simple principles of reducing overlap of nodes and edges, reducing overlap of nodes and crossings of edges usually end up creating a good layout. The most popular type of network layout is, that's the most general one as well that most people use, is called a force-directed network that kind of thinks of tries to simulate the network as a physical system where nodes are repelling each other. Maybe they're like charges that repel each other in physics. And the edges are pulling the nodes together. So they're providing some force that's pulling nodes together. Maybe they represented as springs or some other way of pulling nodes together. And the way that they basically work is simulate this network and then throw the network up in the air and shaking around, and then when it comes down and settles, it's sort of figured out based on all these forces that the nodes are repelling, so they're not overlapping each other, and the edges of kind of pulled nodes that are similar and connected together into groups. And that turns out to work quite well. It's good for networks. It's usually very good for networks up to 500 nodes. Bigger networks you can have problems visualizing them if there's too many nodes and too many edges. So in general, if you have ever a network that you're trying to visualize and it's too complex, well, one, you could try to visualize the heat map version. The other way is to reduce the number of edges that you are looking at. And for instance, in the weighted correlation networks, you could filter the edges based on correlation. So you could select only the strongest correlations to visualize. And that would be one way of reducing the number of edges that would make the networks a lot easier to visualize. And also, the patterns in the network would be more visible. They'd be visible more easily. So there are actually quite a lot of other network layout algorithms. So and a lot of these are available in Cytoscape. I'll show you later. It's always a good idea to try these force-directed layout algorithms first. But if you have other types of networks like trees, for instance, you might be interested in phylogenetic trees, or trees that you get from a clustering algorithm, like hierarchical clustering. And there are layout algorithms for those trees as well. Also, if you want to make a publication quality network, just a couple of tips. It's always good to use these automatic layout algorithms as a first pass. They're very good, but they're not perfect. So it's very useful to manually adjust the layout to further reduce overlap. Maybe the labels on the nodes are overlapping and you want to reduce those. And then you can also export often from most network visualization tools. You can save the network as a PDF and load it into Adobe Illustrator or some other Adobe PDF, or vector graphic drawing program, and adjust the labels and emphasize certain things and highlight certain things, and usually makes for a better network that you, when you're ready to have a publication quality figure. Okay, so I mentioned that bigger networks tend to be complicated to sort of see any structure in, like this big orange one here. This is another sort of general idea just to deal with these networks, is to focus your analysis. If you're ever, so one thing that I mentioned just a second ago is that you can reduce the number of edges in nodes. Reducing the number of edges could be accomplished by filtering if you have a weight on the edge. But for nodes, you generally need to define some kind of filter for what nodes you want to see. So it could be a particular biological process that you're interested in, like cell cycle. Just show me what this network looks like if I look just at the cell cycle genes. And that will generally make the network more visible. Finally, there's also a lot of visual features that you can use to visualize all sorts of types of data on the network, just like I showed with this network diagram I showed earlier. So here we've used node size for the transcriptional amplitude, edge width for the gene co-expression or gene expression correlation as measured with Pearson correlation, and color is set to a particular function, and did I miss anything? So those are sort of three visual attributes that we've used to visualize different parts of this data. And so once you've visualized, and then we applied the force-directed layout, and we got this picture. So, and then actually we loaded it into Adobe Illustrator, and we added things like this little arrow that highlights a gene that we're interested in, and this purple bubble that highlights this module that we might have been interested in, this orange bubble here. So those are examples of where we've manually kind of made changes to emphasize certain things. Okay, so what are things to look for when you're confronted with a network like this? So this is a protein interaction network, but there's actually a few general points that you can look for in any network that you ever see. So one is obviously data relationships. So we see relationships between nodes. It's much clearer here than in other ways of visualizing the data. There's this idea of guilt by association. So which is very useful for gene function prediction. If you have, as that I mentioned earlier, if you have a gene that you don't know what its function is, you can see what it's connected to, and that might give you a clue about it. So that's one type of pattern that's been very useful in networks. Next is dense clusters. So there's definitely a few modules in here. Here's one. Here's a very dense one, and these are fairly visible from once you have a good network visualization or a good network layout. Sorry, I forgot to mention one thing with data relationships. So not only can you see which nodes are connected to other nodes, but you can see relationships between the types of data that I've overlaid on this network. So I've overlaid gene expression data in a couple of different ways on this network. I can see here, for instance, there's a module, turns out it's the nucleosome. All of these genes are expressed at a very high amount at some point during the cell cycle. They're all part of the nucleosome. They're all interacting with each other at the protein interaction level, and they're also their gene expression profiles are very correlated. So this is a really strong module because it agrees across multiple different types of data. So we see a module in protein interactions. We see a module, a known functional module that we've loaded in from a database, the nucleosome. We see the genes are all very similar in terms of their expression, the level of expression, and also the correlation of expression across the time points of the cell cycle. So this is an example of how you can very quickly see quite a lot of strong agreement between all these different types of data that would be difficult to see if they were all separated. Okay, and finally, global relationships might help you see that certain modules are closer to each other than others. So the nucleosome and the kinetochore seem to be more connected than the kinetochore is to the replication fork on this side. And this is not too important. It's not too. It's pretty obvious in this network, but if you have bigger networks, often you can see interesting global relationships between modules. That might help you understand the system. Okay, so quick summary: automatic layout is required to visualize networks, and you can. Networks also help you visualize interesting relationships. You can avoid this very complex networks that people call hairballs by focusing your analysis, and think about visual attributes the use of visual attributes to overlay different types of data on your network at the same time. Okay, so any questions so far? Nope. Okay, so I'm going to move to talking about Cytoscape. So, and I'll talk about network visualization analysis using Cytoscape, and I'll go into a demo and talk about specific types of analysis that you can do with Cytoscape. So Cytoscape is a free software for network visualization and analysis. It was originally developed at the Institute for Systems Biology in Seattle by Trey Eideker and Benner Schwakowski in 2001. And since then, those two people have moved to different places, and more people have joined the collaboration once it became a freely accessible open source project that allows anyone who wants to contribute to it. And so now there's actually a number of different groups, Agilent Technologies, people at Sloan Kettering, the Pasteur Institute, UCSD, UCSF, my lab in the University of Toronto, and others who are contributing to this software. It's probably one of the more popular ones that's out there now, and that's been useful because people have contributed a lot of interesting features to the system. So the idea with Cytoscape is that Cytoscape is like a visualizer. You have to load in data, either network data that you have. So for instance, if you have a correlation network, you could visualize that in Cytoscape. That's where you're starting with a network and you can visualize a network. That's great. Often people have a list of genes that they're interested in, and they want to do some network analysis starting with a list of genes. In that case, you have to get a network somehow. Maybe you can get a network through correlation analysis. You could also get a network by looking up protein interactions and other types of network information from databases, which I'll mention. And then you can load it into Cytoscape, visualize it, and then there's a bunch of types of analyses tools. So Cytoscape, by default, doesn't have a lot of analysis functionality. It's really about network visualization. It has ways of manipulating a network, so you can load up a network and move nodes around. It has a lot of different algorithms for automatic layout. It has a fairly good filtering system that allows you to, once you have your data in the system, to identify, to run queries on it, basically. There are actually a number of ways of getting data into the system, searching different databases, but that's mostly what Cytoscape does. So when you load it initially, you could play with the sample data, but you really do have to get some data into the system, and that sometimes is a stumbling block for new users. Okay, so but the good thing about one good thing about Cytoscape is that because it's one of the more popular tools, and probably because it started earliest and was the first one that was available freely, it's developed a large community around it. There's now, I think, 5,000 downloads a month and tens of thousands of users, and all these people are some of these people in the community are developing tutorials, and they're publishing papers. They're active on the mailing list, and there's a lot of different documentation and data sets that you can use. Also, and probably the most powerful aspect of Cytoscape is that the functionality of Cytoscape is extendable using plugins or apps. We used to call them plugins, now we call them apps, because people liked apps better. So now we have an app store at apps.cytoscape.org. Okay, so there's over 160 now, and they do all sorts of things, which I'll talk about in a bit. Some examples, but Cytoscape's written in the Java programming language, and it's possible to build your own apps. You have to know Java or have a friend who knows Java, and you can, if you wanted to develop new an extension of Cytoscape for a particular type of analysis, you could do that. You could write a plugin, and then it goes on the App Store, and people can immediately download it whenever they have Cytoscape. In the future, we're working towards developing technology that helps people write extensions, that will help people write extensions in other languages like R, Python. So you won't have to know Java to extend the functionality, but that's a little ways off still. Here's just a fun slide of one of the Cytoscape retreats where people were spelling Cytoscape when they, outside of our building. Okay, so so the basic message is Cytoscape is a useful free software tool for network visualization and analysis. How many people have used Cytoscape before? Okay, a few. How many people got a chance to install it on their laptop already? Okay, because we made a link available on the course website. So I'm going to give a demo of Cytoscape now, and this is an opportunity to basically for me to just show you some features so you can get an idea of what it does, and you can ask questions also during the demo or afterwards whenever you see me to ask me if Cytoscape can do something. Can Cytoscape help me with my problem? Okay, so I'm going to be using Cytoscape 2.8.2. This was the last version. The newest version is 3.0. 3.0 is our new architecture of Cytoscape that's going to be faster and have more features. But right now, a lot of the plugins that were in 2.8.2 are not all ported over to 3.0. So I still use 2.8 for demos and teaching because some particularly useful analyses modules are still only available in 2.8 series, although this will hopefully within the next year or so will be changed as all these 160 plugins get moved over. Okay, so when you start Cytoscape, you get a view like this. So I just loaded up Cytoscape by default, and one of the first things that you have to do is import some data into Cytoscape. And there's a lot of different ways of importing data. So in the File menu, if I go to Import, I can import a network the typical way people would do this is import a network from a table, which is a text file like tab delimited text, or an Excel file. The Excel file could have any type of data that you want. It could have interactions, it could have attributes for nodes and edges, so you can import this information. So you can also import network from various web services, so you can search databases, and you can load in ontology information like gene ontology, but I'll just show you initially importing a network. So if you've ever loaded in text files into Excel, this will be very familiar. So I'm going to go to the Cytoscape directory, and there's a sample data directory here. People who have installed Cytoscape locally, you don't have to follow along now. If you want, you can, but it's probably better that you just look what I'm doing, and then you can repeat it later and ask questions if you get stuck. So also, I previously sent a link to a couple of tutorials that go through a lot of this, although I'll touch on some additional parts. Okay, so I'm going to load up an Excel file that's in this sample directory, and this gives me a preview of the columns in this Excel file. And I have to define which columns contain network information. So the source node in this case is in column 1, and so now that's highlighted. The target node is in column 2, and the interaction type, which can be any string, I'm just going to select column 3. So this 3 says PP. That in this example file means protein-protein interaction or protein-protein link. Sometimes it says PD, which means protein DNA. In this case, it's like a transcription factor regulating another gene. Okay, so that's all we really have to do. If there's other attributes here, like this column says true, and it's mostly true, I guess, but it could say other things, then you could import it as well. Oh, here's a false. And you could import it, and it would be an additional attribute on these edges. So each row here is an interaction, and each row is going to be an interaction, and additional columns just have additional information on those interactions, like whether the edge is weighted or some additional information. Okay, so I'm going to import this, and it's imported here. And yep.
Does Cytoscape interpret some interactions as directed and others as undirected by default, or do you have to tell it that after?
So by default, the question is, does Cytoscape treat interactions as directed or undirected by default? So by default, everything is directed, but internally, it's directed. But when you visualize it, you can set the arrows to go whichever way you want. And so if you want to encode direction, a lot of people don't encode direction, and they just load up data in any which way. You won't see direction in that sense being a big part of the visualization. It's settable how you want the arrows to be. So if you do want direction to be encoded, you just be careful to set the source and the target node appropriately, and then you can visualize it, and it will respect that. So here's Cytoscape's initial view. As I told you, if you don't have any layout, in this case, it just shows it as a grid. And then the first thing that you do is go to the layout menu and look through these layouts and try them out. So I like this Y files organic layout. That's my personal favorite. It's a force-directed layout. I can now zoom in. I'm just using my mouse zoom to zoom in here and out, and I can see that it's a lot more understandable. I can see certain modules and certain regions that are more connected. All these guys at the bottom here are disconnected, and so now I get to see some of the structure of the network I've loaded in. So Cytoscape has a lot of different types of layouts. It has a circular layout. It has a hierarchical layout if you have more hierarchical data. And so this is sort of layout acrobatics. But what I encourage you to do is just whenever you load in a network into Cytoscape, you can just try out all the layouts and see which one looks good. It doesn't really matter which layout method you're using. The point is that what you want to get something that's aesthetically pleasing. And some people often ask what the distance of the edge means when you lay out a network. So some of these lines or edges are longer than others. In this case, in the typical layout, it doesn't mean anything. It just the length is just set to connect nodes, and the node position is defined just to deal with this overlap and edge crossing issue. However, if you have a weighted network, you can, there are algorithms, there are layout algorithms. So for instance, one of the Cytoscape algorithms, there's a few edge-weighted force-directed layout algorithms. I like the spring-embedded one. There's edge-weighted spring-embedded. In this case, if you have correlation weights, for instance, on your edges, you can have the layout algorithm respect those, and nodes that are more highly correlated will be placed closer to each other than other nodes. Okay, so another just something that confuses a lot of people is often they see the network doesn't have any labels, and then you zoom in and the labels appear. This is a kind of optimization for Cytoscape. For if you have a big network, it doesn't draw all the details. You can always set it to, and the view menu to show all the details all the time, and then it just forces it to always show all the details. On faster computers, it's fine to do that. This little area here allows me to navigate around the network, and you can move nodes around and select nodes and move them around as a group, and I can do things like rotate nodes, etc. And so you can have fun laying things out, and there's aligning and distributing and scaling, which helps you with manual editing of a layout. So okay. The other thing I mentioned is one thing I mentioned is you'd want to, if you have a big network, you want to kind of focus on some nodes, and if you select some nodes in Cytoscape and you want to focus on those, you can always just create a new network from these selected nodes, and then it just creates that network by itself. I'm going to use this layout button here to lay them out. And so if you're focusing in, you can just create a new network and have multiple networks being visualized. You can zoom in using these buttons or your mouse. And let's see what else. Okay, so the quite a useful tool part of Cytoscape is filters. By default, so let me load up some additional information on here. In this case, I'm going to load up a Cytoscape session file. Because I loaded up some information, it says you can only load up one session at a time. And so I'm going to continue and load up this example in a little bit that has sort of more information in it. I was mentioning earlier that when you start Cytoscape, you import data, networks from tables. You can also import node attributes. But once you're ready to save your work, you save it as a Cytoscape session. And this is a .cys file. It's actually a zip file, so you can rename it to zip and unzip it and see what's inside. But that saves all your networks and your workspace, basically. And so the next time you come to Cytoscape, you can load up that session, and everything you have is there. Network attributes. So you can have attributes on nodes and edges. Attributes are any type of data that you have. So node attributes might be, if the nodes are genes, you might load up gene expression data or gene function information. If you're thinking about edges, you might have weights on the edges, for instance, that you could load up. So again, if you want to load that information up, you can import it, import attributes from a table. I'll just select a file here. Again, that's a node attribute table, and you can select this. This again gives you a preview of this file. So this file actually has all sorts of information about nodes. The first column is assumed to be the identifier, the name of your nodes, and then the additional columns are all sorts of additional, in this file, different types of information for these nodes. You can click on these buttons here to get show additional options. So one of the options that's pretty useful is transfer the first line as attribute names. And so when I do this, so in this file I have column headings, ID, alias, annotation, and I want to set those to be the attribute names. So if I click this button, they all just jump up in here. And so now in Cytoscape, these will be the names. These column headings will be the names of the attributes in the system. So I won't import that right now because they've already been imported in the session that I loaded up. And I can select some nodes. And I have to, it's a bit confusing here because the screen is small, so you don't see any change when I did that. So I'm going to make this bigger at the bottom here. And now you can see when I select nodes, this shows me the attributes of nodes that I have loaded in. And there's a whole bunch here. There are various different types of ways of selecting which attributes you want to display here. Clicking this button here will select all the attributes, and now I see that there's lots of information stored about these nodes in this file. Similarly, you can see edge attributes, and in this case I have just one edge attribute that's useful, which is the type of edge PD or PP. So most of the attributes are on the nodes, and it turns out that there's some gene expression data loaded up on these nodes, and this gene expression data has being visualized on this network already. So I'll show you how that works. So this panel here is sort of the default panel that helps you navigate Cytoscape. The next panels, there's an editor and a filters option, but one of the most important parts of Cytoscape, and probably its most unique feature and most powerful feature for visualization, is this visual mapper. So the visual mapper takes attributes that you've loaded in and visualizes them on the network. So I've kind of showed you this pre-baked example here, so I just want to show you exactly how this came to be. So you can select different types of visual attributes, like the color or width of the edge, the color of the node, the size of the node, the border color. There's more than 20 or so here that you can choose from. And once you've chosen a visual attribute to use, you want to map it from your data. You want to map your data to it. So in this case, I take a gene expression column. So I have a few different gene expression columns here, and I choose one. And then I choose what type of mapping I want. And in this case, it's continuous data, so I'm choosing a continuous mapper. If I had some categorical data on the nodes, then you could choose a discrete mapper, in which case you could select node color based on the type of node. For instance, you had patients with one phenotype and patients with another phenotype, and you want to color them differently. And the nodes represented patients or samples. OK, so I'm going to select continuous mapper, and then this, by default, this is already set to kind of be nice, but by default it will just show you a kind of gray. Let me see. Let me actually delete this, and I'll show you how it looks when you don't have anything. So I'm going to choose node label, node color here. So I double click this to create, to start, and I'm going to select an expression data, continuous mapper, and it remembered my preference that to color nodes from red to green. But you can change these colors here, and now I want to select. I want to have underexpressed nodes being blue, and as soon as I do this, the network updates. So you can see now the nodes are colored from blue to green. So in this case, and I can change these other settings here. So whenever I change these settings, you can see the network changing automatically. So this is very useful if you want to, in this case, I map gene expression data. This is fold change data. So the data was, and it's log2 fold change. So it's centered around 0. 0 means no change. And if this is a change in ratio of gene expression from sample to control. And positive number is big. The bigger the positive number, the more difference, the more higher the gene expression in the sample versus the control, the lower the, and vice versa for lower. So now I have all my gene expression visualized here, and I can zoom out and let me show graphics details. So now I can see what regions of the network are underexpressed or overexpressed. So for instance, this green region here is more overexpressed, and this blue region here is more underexpressed. And it's not very, the colors are a bit faint here, so I could always go back to the viz mapper and change these colors so that I get more deeper colors here for the dynamic range that I've selected here. Okay, so so a couple more things. By default, Cytoscape searches the names of these. If you want to search for things that you've loaded, search your data set, you could use this search box. So I'm going to type in some a search here. I'm going to say select all nodes that start with YAL, and it selects them. They're all shown at the bottom here. This is, by default, it searches this identifier column. Sometimes that column is not that useful, and you want to search other things, so you have to click on this little button here, and then you can search different things, like I'm going to search gene expression. Where is it? Here it is. And it gives you some samples. These are all numbers, like I explained. So now this search box changes into a slider bar. And so now I can just use the slider bar to select genes that have a particular fold change. So I want to select all the overexpressed genes. I just move this like this, and you can take all the overexpressed genes, select all the overexpressed genes. You can do the same thing for edges, and if you had a weighted correlation network, you could use this feature to quickly select and dynamically select only the strongest correlations, and then you could take those and you could move those into a new network, and then that would help you focus your analysis potentially. The other thing you can do is you can just delete those. You could select a set of edges and delete them. That's like all the low confidence edges. So that's particularly useful. And then finally, this filters system allows you to create Boolean combinations of these types of filters. So you can create a new filter on any attribute, and then every time you, oops, I have to select something here. So now I have an attribute on one of these, a filter on one of these attributes. I can add additional ones here. Entree gene IDs that start with 1. That's not very useful, but okay. So and you can make Boolean, sort of arbitrarily complicated combinations of Boolean combinations of filters to search your network. Okay. Any questions? Yep.
Is there a good way to overlay time series data?
Yes. So not in the default version of Cytoscape, but if you go to plugins, you can download plugins from various different sources. And a good one for time series data is called Vista Clara. And I'll show you, I think I'm mostly done with the Cytoscape demo. I'm going to move back to my presentation, and I have an example of Vista Clara that I can show you. Any other questions?
What's the file format? The file format for the input?
Cytoscape recognizes many different file formats. The one that most people end up using is Excel files or tab-delimited files, but it also supports certain types of network files like XGMML.
Project. I use a little bit project. If I write a project file, can you?
A project file?
Yeah.
You mean our project file? Yeah, so if you're working with, so the question is can Cytoscape load up an R project file? Cytoscape, by default, can't just load up the R project file, but because there's a number of people here, this course is mostly focused on R, I wanted to mention one of the plugins which somebody's developed is called R Cytoscape. So what R Cytoscape is designed to do is, from within R, it's a bioconductor package. You have to download a plugin on the Cytoscape side and a package in R from Bioconductor, the R Cytoscape package. And then once you have that installed and Cytoscape is running, any network you have or matrix that you have in R, you can send to Cytoscape and it will visualize it. And then you can use R Cytoscape commands to do all the visualization tricks that I showed you here, but you do them from R. And so that's an interesting way of connecting R to Cytoscape. It's been very popular with people that use R, and actually someone asked about time series. The person who's developed this R Cytoscape is very interested in visualizing time series from within R, and he's created various. He's still working on it, but there's sort of certain functions. They might not be in the latest version, but he's working on a system where you can kind of show animations of your data across a network, like a time series. But let me show you Vista Clara for that as well. Any other questions? Yeah?
Actually, I'm still a little bit confused with the input format. So, so for example, if I have a correlation matrix of, say, 500 genes, and then I have the expression values of each of those 500 genes, and if I have the differential expression values, so how I input this data in a single Excel file?
So
how the columns and rows will look like?
Maybe if I can jump in real quick. So WGCNA has an R command called export to Cytoscape, and it does exactly what it says. So it allows you to export an adjacency matrix into an input file for Cytoscape. I'll show that to you tomorrow.
So I'm just wanted to look how the file will look like what will be the columns and what will be the rows.
Yeah.
Okay, I'll show that to you. So the question is, what does the input file format really look like? So I'll show you an example. You actually need more than one file format. So you need one file for the edges and one file for node attributes. Edges and edge attributes can all go together, and then node attributes. So you need two files at least for that. So the one is that I loaded up here, this Excel spreadsheet, is oops, this looks strange. Oh, there it is. Okay, this is not the right one. This is the right one. Okay, so in this case, I have, this is a spreadsheet that stores edges. So I have, this column is one side of the edge, and this column is the other side of the edge. So if you had correlation values, they could go in another column like this one here. And if you had headings on that, you could call it node A, node B, and correlation. So that would be the way to load up the network. And I'm sure the WCGNA outputs a file that's similar to that. Then the second type of information what you were asking about is gene expression data. So you have gene expression data on nodes. So now I'm going to load up another spreadsheet. In this case, I have my one column is the node identifier, which is the same node identifiers you used for the edge format. You should always use the same ones. And then other columns are additional data. And if we zoom over to this in this spreadsheet, I think this spreadsheet doesn't have gene expression data. It just has gene ontology annotation. But let's try and find one with gene expression data. Here, so here's sort of a typical gene expression data file. This you tell Cytoscape is your nodes, and then all of these could be loaded in as gene expression different samples, for instance. Does that make sense?
Yeah, and so the program will integrate these three files
Yes.
together? So I can open three files at the same time?
No, you load up the network first, and then you overlay attributes.
Over it, okay.
Yeah. So it's all done through the import menu, and so you import a network from a table, and then once you have that network loaded up, then you can import attributes from a table, and you can load as many attributes as you want. Okay?
But in that example, there were three columns that you didn't include in the original upload for the network.
Which example?
The very first network that you uploaded.
Okay, yes.
Right, there were three additional that were going to be blue, right? Had you clicked on them?
Yes.
Could those be attributes so they can do it in one time?
Yes,
Okay.
yes, but for edges. So when you load up a network, the additional columns can be edge attributes. So you can load them all up at once. So that would be useful for the correlation values for edges.
How many files for the network? Is there only one file? Is there only edge or edge and node?
So one file is required to define the network. And then if you want to load up additional data on nodes and edges, you could load up more files. You can store them in minimum 2 files, if you have more files, you can just keep loading up files. So you have to, if you go through the tutorial and try out the sample data, you can see how that works. The tutorial is quite detailed in which buttons to click to load up the data. But this should give you a good. This is the reason I went through it in this level is because when people start with Cytoscape, they don't realize how to get data into the program, and that's one of the biggest challenges for starting up. There are lots of other ways of getting data in. For instance, there's a plugin that we've developed called Gene Mania, and I'll mention it again tomorrow. You can give it a list of genes, and we'll find connections, protein interactions, pathway connections, gene expression correlation connections, and you can just choose which connections you want and load them up. It only supports 7 and soon to be 8 organisms, but it's very comprehensive for those organisms. So that's an example. There's actually quite a few plugins like that. Okay, any other questions? Okay, so I'm going to now skip to a slide that I have here that shows just to reiterate, shows a workflow of how you kind of use Cytoscape. So people tend to start with either gene lists and attributes, or they start with a network. Here are some apps or plugins that handle these things. So you can get attributes from expression data or phenotypes. A network you can get from Gene Mania or IRefWeb or Agilent Literature Search. There's different plugins or string. You might have different types of networks, a correlation network. And then Cytoscape is the part that you use for visualizing it. And you can filter, and then you can, if you want to do analysis, you can download plugins, and they do specific things. Like this Bingo does gene set enrichment analysis, or Go on gene ontology and enrichment analysis, which is a very popular type of pathway analysis. There's also a bunch of different. There's an interesting plugin called Cluster Maker that implements more than a dozen different types of module finding algorithms. So if you're looking for modules and you want to try different algorithms, you can try them out in Cluster Maker. Okay, I'm going to go through just a couple here, and just to give you a sense of some of the ones that I found useful. The first one is VistaClara. It answers the earlier question about a good plugin for visualizing time series data. VistaClara is useful for visualizing gene expression data where you have multiple samples. They could be time points. One of the useful things about VistaClara, and I guess I don't have an example of it here, is that you can, let's see if I can quickly install it and see if this works. When I installed this VistaClara plugin, and you have to look at the manual to see that it puts a little additional panel at the bottom here. And now I can select nodes. I think I have to synchronize this. And now I'm going to make this smaller and make this bigger. So now I can see all my gene expression data here, all my node attributes, and they're visualized here. But one of the interesting things that you can do is enable different kinds of visualizations. One of them is a display heat strips. So I click that, and now if I zoom into this network, I see these little bar plots that are attached to the nodes that show the gene expression data that I've loaded up. And in this case, it's just 3, but now I can see, this would be very useful for time series. You could see different patterns. And in Cytoscape 3, this is actually going to be the next generation of Cytoscape 3. Eventually, this is going to be part of the core system, but right now you have to use VistaClara. So that's one example. Okay, so in these slides, which I've made available, there are, if you want to try out any of these tools, I've added a slide that just gives you some a little lab that you can run through if you're interested in. So for VistaClara, if you're interested in trying it out, you can just follow this little recipe to do what I did just now, and then explore the functions of Cytoscape, of VistaClara. Okay, so the most popular plugin actually for Cytoscape is called Bingo. It calculates does overrepresentation analysis of genes that you select in a network with respect to a background. How many people know about Go enrichment analysis? Okay, how many people have not heard of Go enrichment analysis? Okay, a few people. So the idea of enrichment analysis is that you have a set of genes and you're looking for a particular attribute of those genes that's enriched more than you would expect. So if I give you 100 genes and 50 of them are cell cycle genes, but you know that only 5% of the genome is in the cell cycle, 50 out of 100 is more than 5%, so that's enriched. So half of the genes in the list are cell cycle, but only 5% in the genome. So, and there are a number of different ways of assessing p-values for that type of test, and a number of different types of attributes that people use. Mostly people use gene ontology when they're looking at genes, which provides a lot of different functional annotations for genes, pathways, cellular components, and different types of molecular function. So Bingo does that type of analysis and visualizes the result as on this gene ontology hierarchy, where the significance of the enrichment is colored on the terms, so you can get a visualization of that. There was an older plugin that, question, yes?
The plugin, does it only do Go enrichments, or can I load up my favorite gene sets too?
It can load up Go annotation formatted files. So as long as you make your annotations available in Go format, it could load them up, but primarily it's designed for gene ontology.
So it has to be hierarchical?
It doesn't have to, that's a good question. I think it probably does need a hierarchy. So there are other, I'll actually talk tomorrow about visualizations for enrichment analysis that don't depend on Bingo, and you can use other tools like GSEA or other tools that don't require a hierarchy relationship between sets. So that's a good question. So the question was, does Bingo support other things, other types of attributes other than Go or gene ontology? Yeah. Okay. This is a little bit of an older plugin, but it was really good. So you have to use an older version of Cytoscape to use it. But this was also really good for time series data. So it would allow you to see the network, little snapshots of the network at different time series and show you at different time points and show you expression data over time. It had a really nice, it has really nice functions for coloring and visualizing this type of data. There's also an app called Finding Active Modules from Trey Eideker's lab at UCSD. He published this method quite a while ago, and there's the idea is that if you have a network of gene-gene relationships, often it's protein interactions, but it could be any type of gene or protein relationships, and you have gene expression significance values, you know that a particular gene is significantly differentially expressed in, say, cases versus controls or sample versus controls. And Active Modules takes both of this information together and tries to find regions of the network that are significantly connected and differentially expressed. So this might be useful for finding modules that are really changing their differential in a particular experiment. And you can just run this analysis and it will highlight some modules, and if you click on them, it will select all the nodes that are in that module. So basically, this module that it highlights, in this case, is differentially expressed at these three in these three conditions, and it gets the highest score of all the modules, such modules in the network. So I mentioned network clustering, Cluster Maker. I think I'll skip that because we've talked about it before. Another useful tool is that's quite popular is Agilent literature search. This tool allows you to collect network information from PubMed abstracts. So you give it a set of genes that you're interested in. You put the genes in here. You can include a context. That context can be any word, but people most often use a type of disease like breast cancer or osteoporosis, or and then they there's a couple of other things to select here, like the organism and how many abstracts you want to search. It will search PubMed for abstracts that mention those genes, and then it will look in those abstracts for sentences that have that mention interactions between the genes, and it will look for sentences like gene A interacts with gene B, or gene A binds to gene B, or regulates gene B. It has a dictionary of verbs that indicate interaction, and then it draws the network, and you can right-click on the edges and see the sentences that were used to create that edge. And if you don't like that sentence, you can delete it, and then the edges relating to that sentence will be deleted. Can anyone think of a reason why you'd want to delete? If this tool is just looking for like 2 genes in a sentence and then a term that says regulates, binds, activates, something like that, is there a reason why that might be wrong? Did not, yeah, exactly. Did not find. So inhibits might be still okay, but if it did not inhibit, it might not get that right. So so that's why whenever you do text mining, there's problems like that you have to kind of look at the sentences and make sure that they're okay before you trust the data. But it's quite useful, especially when you can't find a lot of information about genes from public databases, and you want to look in the literature, and there is some information. Okay, I'll skip over this one. The next set of slides is really for your information, and if you're using Cytoscape a lot, you might look through these slides that just talk about tips and tricks for Cytoscape, things that commonly are encountered. For instance, one of the biggest ones is memory. So if you're working with big networks, sometimes Cytoscape runs out of memory, and this is a problem with all Java programs on any computer. Unfortunately, Java sets particular memory, and it keeps that memory. It doesn't have a way of expanding it automatically. So there's instructions for how to increase that. Okay, and then Cytoscape 3.0 is out. You can try it out. It's probably smoother functionality right now, and this is where the new development is going, and a lot more features will be added to Cytoscape 3. Okay, so that's just a quick tour, whirlwind tour of Cytoscape and some of the popular apps. I mentioned there's over 160 of them, so I showed you, I mentioned maybe a dozen. Some of them are not really that, they may be very, very specific, so they're not as generally useful. I focused on ones that I've seen as useful frequently, but there are many more, so I can answer questions about them. Yes?
Please, like, of those 160 apps, what each of them do, like is small dictionary kind of, and what is available? Just to know.
Yeah, so the question is, where do I find apps and how do I find out more about them? So there's this app store, apps.cytoscape.org, which is new, and there's all the apps are listed. You can search for them by categories, and like network generation apps. And here's Agile Literature Search. If it supports 3.0, it gets a little badge. But there's lots of apps here, and you can find them. You can also search, and also you can just look at. Let's see. There's something at the bottom here. There's a wall of apps. So this just kind of shows you all the apps in all the categories, and these are the icons for the apps. Some of them might give you some information, but probably if you can't find an app from this app store. This app store is fairly new, but we're implementing things that you normally see on app stores, like ratings and things like that. So people will be able to rate apps, well, they can already, and so better apps will be loaded up, will be ranked higher. There's also, it keeps track of which apps are getting downloaded, and so the popular apps, you can sort by popularity and see which ones are most downloaded. So that's a useful way of finding out which apps people are using. And then the Cytoscape community has a mailing list that you can email, and if you ever have a question, you can say, is there, I looked through the App Store, I can't find anything that does this. This is what I'm interested in. And we try to guarantee a response within a week. At our weekly conference call, we go through all the unanswered questions and we make sure we answer them. So you should be able to get a response on that mailing list within a week. So don't be shy to email that list.
Thank you very much.
Okay.